I'm Curtis Adams. Thanks for the introduction on where I've come from. So I've been in Vernon for just over 3 years, and really getting the program rolling at this point, I'd say. So I'm going to talk about three of the major projects that we have going on. First, I'll provide an introduction to my team and who I have working with me right now. So I have Gordon Ramos, a technician. He's helping keep us productive and is a big asset in the program. Philip Hinson is a PhD candidate who was down here in the past and did his coursework, and now is up in Vernon working on the organic project I'm going to describe. Jennifer McMillan is here right now and is working on her master's. And Rajan Shrestha, he's also working on his PhD. He'll be coming down here for coursework beginning in the fall, so many of you will meet him. So the research approaches of the program are systems research and crop physiology. So systems research is basically a direct comparison of cropping systems that differ in multiple factors. So it really is in contrast to component research, right? Like where you look at the effect of a particular component of the system on some effect. In this case, you have many things that differ. In my two major projects, they're organized just in this way. They differ in many ways, and we take a lot of measurements to try to see how the systems function differently. So they can differ in things like crop inputs and timing, intensity, varieties, and other factors. So observations usually made over a more extended time frame, and I'll describe that in the context of my own experiments. Crop physiology, that's measurement of crop plant functions and interactions among plant parts. And in my case, we usually look at that in the context of stress or under different management. We also look at ways of determining crop physiological traits, easier and more rapid. So these are the three major projects that we have going on at the moment, and I'm going to go through each of these one by one. So we're doing a lot of work on grain legumes, on cropping systems work, and then on nitrogen fixation also. The major project funded by the USDA is to integrate Guar into wheat cropping systems, and I'll describe what Guar is for the most of you who don't know in a minute. And we're trying to understand limitations on legume nitrogen fixation in semi-arid regions and how we might manage to improve that. We have an organic wheat project going on funded by the USDA's Organic Transitions Program. It's a direct comparison of transitional organic dual-purpose wheat systems and conventional systems. And then crop stress physiology and phenotyping work. We're working with collaborators around the state to develop a ground-based system for cotton phenotyping, and with other collaborators to work on drought tolerance and yield in wheat. Oh, great, I got some funny formatting stuff going on. So grain legumes. So we are trying to understand limitations and management for nitrogen fixation in legumes. So one of the first things that I saw when I interviewed at Vernon was Guar. It's a really interesting legume that I'd never heard of before, and learned about it there, how it's grown just in that region basically, and in other places around the world. Then I ran into Calvin Trossel, who's worked with it a lot, and he told me that when farmers and researchers dig up the root system of Guar, they rarely find the nodules on the roots that are responsible for nitrogen fixation. So maybe I should back up just a little bit and give some background on that. Rhizobia bacteria are present in the soil ubiquitously, almost everywhere. There's some different species in the soil. If compatible ones are there with a particular legume, they can associate with each other, and they have this really amazing symbiotic relationship where the plant provides carbon or fuel to the bacteria, and then the bacteria supply nitrogen to the plant, right? But if those nodules are not present, then nitrogen fixation is not occurring, and they can also be present but not functional. So the fact that Calvin said, we rarely see those nodules in cropping systems in Texas that's a pretty big deal. It means we're not getting the service out of the legume that we would hope to get, right? That's one of the primary ecosystem services that a farmer would hope. So when I looked into this more, I learned that very little science had been done to investigate it, and so I wanted to know more about what are the environmental limitations that we face in Texas for this function, and can we manage to improve the situation, or even do the basic science to know if we even have a problem at all. The paper that I listed up here, this is one study that we published already on this topic that's provided some nice insights relative to guar. Root nodulation in guar affects soils, rhizobium inoculants in guar varieties, and we have a lot of other work that is ongoing and forthcoming. So the work that Jennifer's doing, in fact, is directly related to this topic. She's going to be looking at several legume species and specifically looking at impacts of water limitation, so we can try to quantify just how much water limits nitrogen fixation in these plants, which we suspect is probably the biggest limiting factor. So just a little background to guar, since we're working on it so much. It's also known as clusterbean, and it's a summer legume. You grow it during the summer season, and it originated in India. It's most widely grown in India by far. About 85% of the world's guar is produced in India, and then quite a bit in Pakistan, and the rest is in pockets around the world, like here in the southern Great Plains. There's more than 100-year history of cultivation in Texas. So it started here as like a green manure crop, but it's been grown to more or less extents for different purposes over time. But it's very, very well adapted to the southern Great Plains region. It's very drought tolerant and heat tolerant. It uses relatively little water, and like I've been talking about, it fixes nitrogen. So that's what guar seed looks like. I was amazed when I first saw it because it just looks like little stones, right? And they're incredibly hard. If you bust those open, you'll feel a sticky gum inside of there. And that gum is called galactomannan gum or guar gum. If you read food labels, you've probably run into that, right? Because it's used as a binder and a thickener and for other purposes in many, many, many foods. It's also used in oil well drilling, in cosmetics and paper manufacturing, and many, many other things. So the United States imports more than $1 billion in guar every year. We have imported up to $3 billion in guar in a single year before. So we're the #1 consumer in the world, but yet we produce just a tiny fraction of the world's guar. So there's potentially an economic opportunity there for us to produce more of this thing we're consuming so much. The place that we use it the most, interestingly enough, is in oil well drilling, not in the food additives or other things. It is used in the process of fracking oil wells. So India and Pakistan, they do exert control on the market to a really great extent and keep prices low, and that's kind of a problem for us. So I think progress in the U.S. guar market really depends on innovation and doing things better than they do, right? Producing it cheaper or other things like that. So that leads into the project, the USDA-NIFA-Funded project. I have all these collaborators working on it on different aspects of it. Calvin Trossel, who I mentioned from Lubbock, and Paul DeLon working on soil aspects. Yubing Fan is working on economics. Srini Ali is in the Vernon Center. He's working on a modeling component. And Garrett Hugenboom and Ken Booty are at the University of Florida and also working on model development, which I'll talk about in a second. So we have 2 field locations, and we're doing a comparison of systems. So we have integrated wheat guar systems that vary in cropping intensity, all the way from broad fallow periods in between to very intensive. So we can get at what would be an optimal intensity to bring these 2 crops together, and comparing that to continuous wheat and continuous guar. So we're doing work in the areas that all those collaborators work in, right? So crop production dynamics, soil nitrogen and carbon, guar model development and simulation, and economics. And we expect a variety of benefits to come from putting these 2 crops together in a cropping system. So the data that I have over here, that's some preliminary data that I included in the proposal for this project, and it came from unrelated research, like where we were doing rotations of cotton with alternative crops, each of these, guar, sesame, and sorghum. And we had cover crops in that system, and in that particular year, it was wheat. And so we had this opportunity to see, guar, cotton, sesame, and sorghum coming out, going out of those things and into wheat, and then what the response was. So the top graph was wheat biomass productivity following each of those crops, and the bottom one is the relative chlorophyll and stad units. So you can see the obvious contrast. The wheat cover crop is doing far better following guar than it was all the other things. That was part of the thing that spurred us to develop this proposal, because we'd seen, we saw this data and we'd seen other data, and the nitrogen benefits and other potential benefits seem to come together to produce a better effect to rotate these 2 things together. So we expect improved and stabilized yield over time in an integrated system, higher precipitation use efficiency, especially if the optimized system can reduce the incidence of fallow, having less fallow in the system. We expect there to be more nitrogen in the soil, improved nitrogen cycling, and increased soil carbon and health. So this is a really big component of the project. There is no existing model for guar, and so through the project, we're going to develop one. It's going to be a DSAT model. The collaborators in Florida are helping to establish the thing, to build the backbone of it. And then we're going to take all the data that we have from the experiment and do simulations. And we're especially going to try to better identify the role that this crop may play in responding to climate change in this region. Okay, I guess I shouldn't say this region. I actually live a long ways away from here. That region. Okay. Okay. All right. We'll shift gears into into our organic project. As I was developing this project, I learned that Texas is behind in organic agriculture. So this map, which it comes from a 2014 publication, it shows the location of every organic farm in the state of Texas, right? So there were 305 organic farms in Texas in 2014, which sounds like a decent number until you consider how many farms there are in Texas, right? So it's a smaller percentage than any other state in the nation. In 2014, only 60% of those farms actually sold goods also. So in our project region, which is Extension Districts 1 and 3, outlined in red there are only 15 organic farms of any kind, despite a lot of potential to grow things organically there. So the Southern Great Plains region, which is really like the Vernon area, Rolling Plains, the High Plains, up into Oklahoma, and some pockets in the area, are very well known for their dual-purpose wheat systems, which is a really amazing and interesting system. It's where you plant wheat relatively early in the fall. It grows up and is established, and then if the weather conditions are right, you can turn cows out on there, and they'll graze it throughout the entire winter. You need to get the cows off before jointing, And if you do, then it'll also produce a crop of grain. So that's the dual purpose, right? You have forage and you have grain production. All these things are made possible by the climate of the region, where it can grow over the winter and continue to produce that forage, and the properties of wheat itself, where it can be grazed and still regenerate. Right? So that's the system that we're studying in this project. So part of the justification for doing it was just the basic demand in these sectors of the economy. The organic wheat, that sector has grown by 9% per year in recent years, and buyers have trouble sourcing enough organic wheat to meet their demand. Texas is a top wheat producing state, but we have hardly any organic wheat farms, so there's a lot of potential there. Organic beef is growing by even more, 20% per year in recent years. And the U.S. definitely doesn't have enough supply to meet its demand. A really large share of our organic beef comes from Australia. So when I think about that, that just doesn't seem to savvy very well, right? Like, why do people buy organic? You know, it's because of like the safety of the food or the sustainability or other things. I don't think that they want it coming, you know, across a barge across the whole world from Australia, right? And when you think about it, like Texas has more beef production than any other state in the nation by quite a bit. So Texas is poised to meet that demand. It's just at this point we're not. And as I've talked with people who are really involved in this stuff, they'll have consensus that it really just has to do with the lack of a processing facility in the state. And so now that our project team is a little more savvy on that, we're starting to do some work to put together something that would better characterize that limitation and understand it, so that information can be out there. Because really, among all the states, Texas is better poised to take advantage of the opportunity than any other place. So the project that we have is, this is the rough title of it, Introducing Organic to Wheat Producers in North Texas. Dr. Bill Pinchak is taking care of all the animal-related stuff on the project for us. Anil Somanhali is working on soil microbiology, Yu Bing Fan on economics, and Emmy Kamira and Jordan Bell on extension. So I mentioned this before, but we're testing the 3-year transition period into organic certification. So if you're not aware of that, like when a farmer decides he wants to go organic in his farm, like they have to shut off all the chemicals and all the fertilizers and the other things that are not compliant with organic, and then wait for a 3-year period before it can finally be certified. And so that's the period that we're studying in this project and directly comparing it to a conventional system. So this is taking place at a really large scale. I did my PhD working on microscopic algae in tubes, tiny tubes, and now I have this experiment that's 100 acres. So it's a really awesome experiment. This facility is called the West Walker Farm or Ranch, and this section of land is divided into different paddocks. We have 8 of them. 4 of them are transitional organic, and 4 of them are conventional. And just like the systems research I talked about in the past, like they differ in many factors. So the organic system, we have crop rotation of wheat and mixed forage. The mixed forage includes legumes to try to bring the nitrogen into the system. So we're going to have biological diversity there, economic diversity, and hopefully nitrogen fixation. We have a summer cover crop in the system to bring more nitrogen fixation, weed suppression, and soil health. We have compost application to bring minor nutrients and other needed nutrients, which we also hope will improve soil health, and tillage to control weeds. In the conventional system, it's being managed in exactly a way a farmer would manage their system there in the area. So crop monocropping, wheat year after year, which it truly is representative of the way that they do it. It's rare to diversify those systems. A summer fallow period, inorganic nutrient supply, tillage, and chemical weed suppression. So these are some of the key things that that we're looking at. Because of the really large scale of the experiment, we have the ability to get reliable information on cattle weight gains by system, and have those cows in there for the full duration of the experiment, over the time frame when they should be there. So we can gather information on that. We're getting information on soil microbiology and health and greenhouse gases, information on cash and cover crop performance, especially forage productivity and quality, wheat yield and quality, and then we're going to be assessing the cover crop for just how well it's doing, how much nitrogen we're getting from that, and so on. And then we're going to look at economic outcomes, because the transition period it's so critical for a producer, because that's the period where they have increased inputs because they have to do things like put on expensive compost and start cover cropping, and then there are a variety of other things that increase their costs during that transition period, but they can't receive the organic price premium because they're not yet organic. So doing economics to really fully help people to understand that issue will help them to overcome barriers to adoption. Okay. So finally, I'm going to talk about our work on crop stress physiology and phenotyping, work funded by Cotton Incorporated and Texas Wheat Producers Board. So this project, the Cotton Project, have collaborators around the state, Nithya Rajan from here on campus, Glenn Ritchie in Lubbock, and Carlos Fernandez in Corpus Christi. And the goal is to develop a ground-based phenotyping system so it would hasten the development of improved and adapted cotton cultivars, right? So rather than doing the intensive kind of measurements they do now, instead they'd be able to deploy this system of a bunch of sensors across the field and hopefully gain information in a more rapid way so they can screen more germplasm. So the system has spectral canopy indices like NDVI, NDRE, and the capability to take others, height and temperature, all those things are sensor-based. And then we're taking additional information on leaf area index and yield so we can relate it to those things. So I included just a little bit of data here. We collect a lot of data in this project, so these are just some examples. The relationship between NDVI and canopy height, for example, is almost a perfect relationship, an R squared of 0.91. Very, very good predictor of plant height in cotton in a ground-based system. This other data here is temperature, and this is a function of time. In the experiment, we had different irrigation treatments, and we also had 8 different varieties, and so all those are averaged to give that graph. Early in the season, we saw, let me describe this also, I guess, 0 on this, because this is the canopy to air temperature difference. So if the point falls above the line, that means that the canopy is warmer than the air, right? If it falls below it, then that means the canopy is cooler than the air. And the idea would be if the canopy is relatively cool, that means that it has access to water, it's transpiring, and transpiration is directly related to photosynthesis, right? So if we're comparing among ET treatments like this, like in a dryland situation versus a very high level of water, we expect to see a trend exactly like that, right? Where the well-watered one can maintain a cooler canopy because it's transpiring, and that's what we saw. But then also, when we looked at it among varieties, there are also differences, differences at key times, and in some cases across the entire season, where we see that one genotype seems to have more access to water or is using water more efficiently and has a cooler canopy. So we're working through the data, but that seems promising that this might be able to separate more promising germplasm from others. Okay, and finally, working with Dr. Shua in Amarillo and Dr. Dong in Uvalde to evaluate crop physiology and crop traits in wheat. So I really like these studies because we look at, we gather a lot of data over the course of the season, and on the development of the crop and when it transitions from one stage of development to another, how much biomass is there, where the plant is putting its biomass, and many other things. But I think the real strength and power we have here is that we have 3 locations and we have a lot of varieties in common. So hopefully we can derive more information on the way these things interact across different environments, genotype by environment interactions. I just wanted to bring up one example that I think is really interesting, because this is the kind of thing that we're after. So TAM 112, it may be one of the more famous wheat varieties in Texas, and that's just because of its really amazing drought tolerance. So it really performs well in drought conditions in the high plains. When you bring it to the rolling plains where I'm located, it doesn't even do so well. But last year we had a really severe drought. It hardly rained at all from October 2017 to October 2018, and then it rained really a lot. When it was not useful for, or when it was useful for the next wheat season, but not the one that had already passed. So at the Bushland station, TAM 112 yielded 30 bushels per acre in that situation where there were hardly water inputs at all, where most other varieties yielded less than 10 bushels per acre. And that simply is due to traits it has, where it's really able to perform well under severe drought conditions, and that's the kind of information we're after. What exactly enables it to do that, and what kind of traits would a breeder need to select for, so they can select for varieties that do well under that condition, or ones that do well under higher water conditions, or ones that are more adaptable across many conditions, and those kind of things. Okay, thank you very much.
That was plenty of time for questions.
Yeah.
So you had a record correlating NDVI with high.
Good.
Yeah. I would like to know if the NDVI was at which time of the season and if you were considering the ground.
That's across the entire season, and that's across every treatment that I had. So that's across many genotypes, all of these irrigation treatments across the entire season.
The NDVI was collected with a handheld?
No, it was with the crop circle sensor. Are you familiar with that one?
Okay.
So it's sort of like a handheld one, except it was integrated through a data logger. So it was on like this rover mounted above the canopy like this, and controlled by a data logger as we rolled through the field. Yeah. Yeah.
Sir, you have a project with the organic practitioner system. So I was wondering how expensive is the documentation within that 3-year time span for a farmer to get certified as an organic?
What was the word you said? How intensive is what?
How expensive is the documentation process for the transition from conventional to organic?
It's kind of intensive. Like a lot of people actually get consultants to help them, I guess, to make sure that they dot every I and cross every T. Just you need good documentation on, when you cut off chemicals, how you store things, and just a lot of factors. I talked with one of those consultants only a couple weeks ago, actually, who helps to shepherd people through the process. So it can be daunting for some.
It consumes a lot of time with this transition, so to document them
It can. I think the biggest trouble is just making sure that you do document everything you're supposed to, and then having knowledge of what you should do. Yeah.
So you mentioned the lack of a processing being a big driver for the low adoption of organic beef. Are there drivers like that for the wheat, on the wheat side that you see, or you just, we don't do it and we never have?
Not so much actually. Like one of the biggest buyers of organic wheat is Ardent Mills. They're based in Colorado, but they're willing to come and to bring it, you know. And there's Arrowhead Mills here in the state, and there may be others I'm not thinking of. But it doesn't seem to be the same kind of limitation, and I can't tell you every reason why that is. Other than, like, I know that the closest beef processing facilities, like there's one that does basically hamburger in Colorado, and that's not really what a farmer would be after, right? That's like a lower value application. And then the other closest one I think of that does like whole cows and all the more valuable cuts of meat is in Iowa. So that's a long ways away, and it's a long way to ship beef, and then the price premium starts to become eroded by shipping them that far. Yeah.
What is the yield trade-off for the simultaneous grazing and grain harvest of wheat? I've never heard of it before. I was just wondering.
Dr. Redmond's way more qualified to answer that than I am.
The work that we did showed that for every day that you go past the jointing or peak stage 6 is about the same thing. It was 1.25 bushels per acre. So, and the problem there is that even if you have the same variety, the jointing time can vary as much as a month from year to year. And so we published a paper talking about looking at first hollow stem. And so you'd find an ungrazed plant somewhere in the corner of the field and cut that off at the ground, slice the stem, and look for evidence of first hollow stem. And so if you see first hollow stem, and then you make your plans to get the cattle out there quickly, you didn't penalize yourself. But if you did, you could penalize yourself tremendously.
So grazing up to that point has no
effect.
That does interact with environmental conditions somewhat, right?
Yeah, and varieties are different too. I mean, you just have to look every year. A lot of farmers say, we pull off at March 10th. Well, you may have already penalized yourself half your yield.
That's basically what you're doing at that point. You go past jointing, the cows are eating the head, you know, that's the primordia of that head. And so it's going to be like a direct yield cut for every one of those they eat. Yeah. Yeah.
How much guar is being grown at this time in the Vernon area? I know 50 years ago there was a processing plant in Vernon, and guar would be used if your cotton crop failed. You could plant it mid to late June and still get a crop, and they would truck it in 50, 60 miles away to that plant.
Yeah.
And then I understand the plant went out of business or burned or something. Is there a processing plant in Vernon now or in that region?
So I can't answer your question on the number of acres exactly, but it's still grown in the Vernon region, a fair amount of it, and all across through Lubbock. The processing facility thing and the companies, that's a very complicated history. So the company that was there in the past is not in business anymore. There's a company in Brownfield, I think it's called Guar Resources, and that's who's contracting guar acres today.
So they have to transport it clear to the high place.
Yeah. Yeah. Dr. Wallensberger, do you have a question?
I was just curious as to what your future vision of guar was. It's a very productive plant in that arena.
Where do I see it going for the farmers and for the state? And it is complicated. Like the way that I mentioned the way that foreign countries, especially India, can exert control over the market, and that can be really damaging to farmers here. But the crop has so many benefits. You mentioned it. putting it in as a crop to follow a failed crop, and that really is where it primarily is utilized. But I think it has so much potential. It uses relatively little water, it fixes nitrogen, it does all these things that if we could do things that elevated its state as more of a cash crop, it might be able to take a bigger role here.
Sure, that's that. So when you were talking about nitrogen fixation capabilities, but were you also saying that we don't see nodulation very much in Texas, or was that for a different crop?
That was for guar.
That was for guar.
But I'd say those assessments are very unscientific, and that's someone,
Okay.
that's someone with a shovel.
Oh,
And
okay.
hoping they find nodules,
Oh, okay.
And like there aren't good published studies on it. At the same time, like I've found tons of literature that talks about the limitations on nitrogen fixation due to water, and we would of course have that. You can take your temperature sensor out to burn in Texas in the middle of the summer, and the surface temperature is 115 degrees or more. So like those nodules are really sensitive to temperature and to heat, and so I'm not surprised you wouldn't find them in that baking soil surface temperature, in the baking surface soil. But what about below that? And, how much does it depend on when they receive water and those kind of questions?
I would mostly think that the mechanisms would be the high heat and the interaction between the heat and the water, and not anything to do with, do you guys fertilize guar?
No, nobody fertilizes guar.
Oh, cool.
Nobody fertilizes guar. Jennifer's studies are going to touch on exactly those questions. We want to know like where are nodules formed and how does the time course of that run? When do they start to senesce? Are they far down? Are they close to the surface? And other studies are helping to supplement that, so we can say, could you water a little bit to rescue those, I guess, or other questions. Yeah.
Can you eat guar?
Yeah. So there's a bunch of different kinds. My old advisor from Florida actually sent me a text message from some market in Gainesville, Florida, and there was the vegetable form of guar there. I haven't had it.
You haven't eaten it?
I haven't eaten it, no. But the industrial type, that produces guar gum, can't eat that. That'll tie up all the water in your stomach. It wouldn't be comfortable to eat that. So,
But we do eat a huge amount of guar, yeah.
yeah, we eat it.
It's in almost every soup.
Yeah, but it's diluted down.
Yeah, I think that's one of the reasons they use it is because you can use minuscule amounts, and it's such a powerful binder and thickener. Yeah.
Upon the industry guar, after using ejected guar, has anybody did any environmental study? What's the environmental fate of this guar? Are they protein or some other, what kind of polymers are they?
No, but that's why they use it, because it's biodegradable and not harmful to the environment.
The people,
In the fracking wells, that's what you're saying? Yeah. Basically it biodegrades and it's not an environmental contaminant. It also works extremely well. Like when prices went up for guar gum, this happened some years ago, they said the drilling industry was looking for alternatives to guar, and they were looking hard, to find something better, and couldn't find something that was as good. So it also was functionally really good, but I don't think they expect it's an environmental contaminant. That's one of the key reasons they use it. Do you have a question? No? Yeah.
I was curious about the guar nodulation. I worked with Dr. Lon, and sometimes it tells me that the aquifers that supply the water for irrigated land are high in nitrate, and if legumes are receiving nitrogen, they won't fixate, or they won't feel nitrogen fixation. So I was wondering if, you believe you said we're digging the shovels, if they were also irrigating as opposed to like dry land.
So I said nobody fertilizes guar, nobody waters guar either. so we don't have to worry about that, but the issue you're talking about is true. And that's true in the Vernon region. We have some of the most elevated groundwater nitrate. Yeah. Yeah.
Are there rhizobia that are specific to guar where you could inoculate the planting, and have they done this?
Yes, so that's the case with every legume. like, if you go to inoculant companies' websites, right, they have products specific to everything. And sometimes, like the inoculant that works for cowpea, for example, works for mung bean too. But there are some that are very specific. And you won't find, among all those inoculant companies, you won't find guar on any label, because none of them are compatible. But in the USDA rhizobia bank, there are 4 for guar, and we have those in hand. We're going to do some testing on those.
But they're not available commercially to someone who wants to grow it, inoculate the seed yet.
No, we have a microbiologist who made them in his lab for us. Yeah. Okay. Yeah, great. Thank you very much.
Thank you.